Good evening. Good evening, participants. My voice is audible to all. Please reply first. Good evening, participants. My voice is audible. Please comment. Okay. I am Dr. Anand. I've completed my PhD from IIT BHU. So I will continue your Proteomics course because Dr. Simran is not well. He is unfit. That's why I will continue this session. Okay. Now my voice is audible. Is it clear to all? If not clear, please tell me. Please comment. Okay. Uh, Please subscribe my channel first. After that, we will start this session. Okay. So please subscribe first. So this session I will continue. Okay. Now we are going to start. So today we will uh, learn about the techniques of proteomics. Okay. So PPT is visible to all or not? So first we will understand the definition of proteomic. proteomics and proteome. Okay, so proteome is the set of all expressed proteins in a cell, tissue, or organism. Our body is made up of protein, lipid, carbohydrate, nucleic acid, but in all body, a set of protein means all proteins will be considered as a proteome. Okay. Now next, the proteomics. Proteomics is the study of proteins, means all the proteins present in a body. Okay, their roles, their structure, their localization, their interactions, and their factors. Okay, we will study under the proteomics. You know, lots of proteins are present in our body. Okay, you expect more than thousand, more than lakh, more than crore type of proteins are present in our body. Okay, so all the proteins forms with the help of genes or nucleic acid. Whatever the nucleic acid is present in our body, okay, different types of proteins will form according to the nucleic acid. If nucleic acid is not present, that protein will not be expressed in our body. So proteomics investigates on a large scale of proteins expressed in a given organism or biological system. A top-down proteomic approach aims to identify characteristics of whole proteins, while bottom-up proteomics, also known as shotgun proteomics, uses a high-throughput workflow to analyze complex mixtures containing hundreds to thousands of unique proteins. Okay. Next, large amounts of proteomic data is increasingly being produced, often as a complement to genomic results, as proteins are the functional macromolecules directly driving cellular activity and physiology. Proteomics offers significant biological insights not readily addressed by genomics and transcriptomics. These include studies in post-translational protein modifications, spatial and temporal protein localization, the biochemistry of metabolic pathway, protein interactions, and more. Okay. So here now we will discuss the tools in proteomics. What type of tools we will discuss? We will give I will give you a brief idea about the tools of proteomics. Okay. So now my voice is audible, or you are facing any problem? Okay. Who have not subscribed my channel, please subscribe first. Okay. Now here we will divide the different phases, okay, of proteomics. First is cell lysis and extraction. Okay, how we will lyse the cells and how we will extract the proteins. Okay, so we will study in this step. Next, cell fractionation is the second step, how we will fractionate the different proteins present in a cell. Next is protein concentration and quantification. Okay, how we will concentrate and quantify our protein present in a cell or body, tissue, anywhere. Okay, next is protein digestion. Okay, next is peptide cleanup and mass spectrometry. So first we will talk about cell lysis and extraction. How we will lyse the cell? Okay, you know, uh, cell membrane is made up of protein. Okay, in the cell membrane different types of components remain present. Okay, you know, lipoprotein, glycoprotein, carbohydrate means polysaccharides, lipids, different types of components remain present. So we have to break these components. After that, all the remaining components will be free, like cell organelles, and all the cell organelles will have proteins. So we have to extract all the proteins present in a cell except other components. So from the cell we will lyse by a lysozyme. You know about the lysozyme? How many of you know about the lysozyme? Please comment. Please comment. How many of you know about the lysozyme? Nobody knows. Okay, lysozyme is an enzyme which will break a cell membrane. Okay, and present in tears or egg white. Okay, you know, one person know, Panathi Ganti. Okay, good. So next thing is this is the lysis method. Some more lysis method we may use, like mechanical. Okay, so by pestle motors or sonication we can break the cell. Okay, some chemicals we may use for the cell lysis. After that we can extract our protein by different types of chromatography technique. Okay, so in detail we will discuss about the chromatography technique. Okay, so before going to chromatography, okay, we will discuss here analysis. Okay, we will perform the analysis by using SDS page. Okay, how many of you know about the SDS page? Please comment. SDS page. I'm not talking about the lysosomes. Okay, I am talking about the lysozyme. Okay, two people know about the SDS page. Okay, three, good. Okay, SDS page is sodium dodecyl sulfate, polyacrylamide gel electrophoresis. So this is the technique in which we can separate our proteins on the basis of molecular field, molecular weight in the electric field. Okay, so how we will define SDS page? This is the technique to separate proteins on the basis of molecular weight in the electric field. Okay, we will learn about the SDS page later. First we will discuss about the brief idea. Okay, next is cell fractionation. After extraction of proteins we will fractionate proteins. Okay, after that we will go for the concentration, concentrate the proteins and quantification. Different methods of quantification are there. UV spectrophotometry we will use here, and after that protein digestion, peptide cleanup, and mass spectrometry. So in a cell lysis and extraction, cell lysis that proceeds protein extraction can occur mechanically through homogenizer or sonicator. I have discussed this thing, or chemical solubilization by detergents or lysis buffer. Okay, while mechanical disruption offers an advantage of maintaining a relatively native chemical environment, reagent based lysis via detergents or lysis buffers are generally more accommodating to smaller sample volumes and higher throughput processing. Ready to use protein extraction kits can offer a rapid, straightforward solution for lysing cells and subsequent protein collection. Okay. Next point is cell fractionation. How we will fractionate the proteins depending on the aim of the study. Complex samples such as whole tissue and cell extracts may require additional fractionation. This step can ensure enrichment of proteins enriched in subcellular locations such as membrane proteins or nuclear proteins. Highly abundant proteins can also limit peptide identification during LC-MS analysis. Okay. Further, fractionation through the use of centrifugation, means ultracentrifuge, chromatography technique, or protein concentration kits can help to account for the variations in protein amounts and to effectively capture lower abundance of proteins. Okay. Now, protein concentration and quantification. Different protocols are available, and different protein concentration kits are available in the market. You can use for the concentrating our protein. Okay, protein digestion to generate peptides for mass spectrometric analysis in the solution, or in gel digestion are the two common strategies. Within solution digestion, proteins can be denatured, digested, alkylated, and reduced in a single tube, making this optional idea for less complex, detergent sensitive, or low volume samples within gel digestion. Protein electrophoresis, typically SDS page, is used to first separate and denature proteins, which are then digested while embedded in a gel slice. Gel based digestion is widely used in LC-MS MS analysis of complex samples. The choice of protease may vary depending on the nature of sample proteins, trypsin being the most common. Okay, so this SDS page will separate your protein. Okay, why we are using SDS? SDS is a sodium dodecyl sulfate. It will denature your protein and it will convert your secondary, tertiary, and quaternary structure of protein in a primary structure, because it have both the charges. Okay, and your protein have both the charge. Maybe it may be negatively charged, positively charged, and it may be amphoteric. Okay, okay, I'll tell you protein concentration kits. Okay, good. Yes, I'll provide you. So, STS page, STS uh, will provide a negative charge to all proteins. Okay, that's why your protein will be negatively charged. It will move towards the positive charge. Okay, on the basis of molecular weight. Now, next is the peptide cleanup and mass spectrometry. As detergents and other contaminants obscure mass spectrometry readings, peptides must be cleaned prior to analysis. C18 reverse phase is the most commonly used liquid chromatography stationary phase region for the cleanup and can come in column, cartridge or pipette tip formats. MALDI is a matrix assisted laser desorption ionization and ESI electro spray ionization are the two choice ionization methods leading to up to MS and MS analysis, with LC-ESI MS systems being favored for complex samples. Mass spectrometry services that specialize in proteomic analysis are also available. Okay, these are the very sophisticated instruments, and for the peptide study we will use this. Next is we will hear uh, study about the molecular techniques first, then separation techniques, next protein identification techniques, next protein structure techniques. Do you know about the structure of proteins? Different types of structures are. Please comment. Have you basic idea about the amino acid? How many types of amino acids are there? Please comment. Please comment. Amino acid sequence, peptide sequence, and basic structure of proteins. Primary structure, secondary structure, tertiary structure, and quaternary structure. And homology modeling. Okay, some people know. Okay, good. Essential, non-essential amino acid. Okay, there are generally 20 amino acids. Okay, okay, good. Homology, not much, sir.